doing. So again, welcome to our Turing-Roche Knowledge Share Series event. The aim of these monthly events are to bring together members of the Turing and Roche networks and the wider scientific community to hear partnership updates, different perspectives on topics in data science, and also build new connections. I'm just going to give a quick overview of the partnership via some slides. Hopefully you will all be able to see that. So the clue is in the name of the partnership. It's between the Alan Turing Institute and Roche. Hopefully most of you are familiar with at least one of these organizations. The Turing Institute is the UK's National Institute for Data Science and AI. We have a research in a number of relevant programs, and Roche is our first pharmaceutical partner. And Roche established in 1896 as a leading healthcare company, and also have a growing number of academic partnerships. And we launched our 5-year strategic collaboration in June 2021. And this is our partnership North Star, which is our kind of guiding light over the 5 years. So ultimately, we're kind of collaborating to explore this term patient and disease heterogeneity, doing this through advanced analytics. So why patients respond differently to treatment and why diseases manifest in different ways in different people. Obviously, this is quite broad, so we're kind of tackling this through a kind of stepwise, iterative manner, which you can see illustrated on the right-hand side of this slide, with a main focus on kind of developing methodology, and we're also focusing on different research themes such as structured missingness and predictive modeling. You can find out more about this on our Turing-Roche webpage, which is on the Turing website. Excuse me, just got a bit of a cold. So our core partnership team is pictured here, and this is expanded out with researchers we have funded. We've also taken on board Turing enrichment students. And again, you can find more detail about who's involved on our webpage. And a key part of the partnership is kind of wider connections and collaborations we want to build. So it's really critical for us to be running events like these to get you all involved. And I know some of you have attended our events before. So our normal format is having a Turing speaker and a Roche speaker talking about different data science topics. And we will be continuing this for some of our events, but we're also really excited that as the partnership research is progressing, we'll be shining a light on that at some of our events as well. So in that vein, I'm delighted to welcome our speaker today, Chris Banerji, who is a senior research associate for the Turing-Roche partnership. Our setup for our senior postdocs is 50% partnership research, and 50% their kind of own independent research. Obviously, there's a lot of overlap, but we're really excited to have Chris here today, who'll be talking through some of his research from his work at King's College London around understanding multi-scale heterogeneity and the impact this will have on personalized medicine. So with that, I'm going to stop sharing. I will hand over to you.
Lovely, thank you very much, and thank you everyone for answering that poll. Right, I will share my screen and begin the presentation. Right, so as Vicky said, my name is Chris, and thank you very much for coming here today to attend my knowledge share as part of the Turing-Roche partnership. As Vicky said, I'm one of the senior postdocs at the Turing, but I'm also a clinician. And today I'm going to be sharing with you some work that we've been doing over the last few years, focused on this concept of multi-scale heterogeneity and the implications of this multi-scale heterogeneity in complex pathology, and really trying to improve our understanding of how to develop therapies and also optimize the clinical trial landscape in rare and complex disease. And I'll be using one particular disease in particular, called facioscapulohumeral muscular dystrophy, which is a rare disease that I really think is a poster child for this problem of multi-scale heterogeneity. So what do I mean by multi-scale heterogeneity? Well, ultimately, it's a concept that probably many people will be familiar with, but actually has manifestations at deeper scales than one might imagine. So the main scale that people tend to look at is this scale that is the population scale. So this is looking at different people, all of these different people here, who all have the same pathology, but might manifest that pathology in different ways as a consequence of their genetics or their inherent risk factors. And as a consequence of this, these different individuals might be expected to have different disease severities or perhaps different presenting symptoms in their pathology. And understanding these differences in the same disease and how it manifests across different people is this problem of interpatient heterogeneity. It has a lot of ramifications for clinical trial stratification and other sorts of analysis of patient populations. However, if you were to consider just one individual from that population and look to see how the disease manifests across that one individual, you might find that in different tissues, you have different representations of the disease burden. So if this was an inflammatory pathology, you might find that some tissues have a higher level of inflammation than others. If this is a malignant pathology, you might find that some tissues have a higher disease or tumor burden than others. And understanding this heterogeneity within the individual, this intra-patient heterogeneity, also has a lot of ramifications, particularly when we're considering biomarkers of disease progression, such as imaging biomarkers or biopsy-based biomarkers. But it doesn't stop there. If you were to take a single tissue from that individual, let's say one that has high disease burden, and examine the molecular landscape of the individual cells within that tissue, you might find that there is a huge heterogeneity in the level of molecular markers of treatment resistance, disease progression, or even the presence of your drug target in particular. And this diversity within the tissue scale, this intercellular heterogeneity, has huge implications when we're considering residual disease burden after treatment and even the efficacy of our therapies that we develop. So hopefully I've convinced you with this little cartoon that there are many different scales of looking at heterogeneity in complex pathology. And I would argue that understanding all of these different scales of heterogeneity are really important when we want to understand how to clinically improve the situation for patients with complex disease. And as I mentioned before, understanding heterogeneity at this population scale, this interpatient heterogeneity, can give us an understanding of which patients are going to be fast progressors and which patients are going to be slow progressors. And this has huge implications for clinical trials where you want to make sure that you equally allocate these two patient groups to your treatment and control arms so that you do not mask a potential impact of therapy. At the individual scale, understanding this intrapatient heterogeneity, as I mentioned, is crucial for understanding tissue-based and imaging biomarkers, because there is a huge variability in how the disease manifests within a single individual. This needs to be taken into account when you're monitoring them using these biomarkers. And as these biomarkers are increasingly being used as surrogate outcome measures for clinical trials, we really need to understand heterogeneity within an individual to be able to use them properly in this circumstance. And of course, as I mentioned, the intra-tissue scale heterogeneity in terms of the expression of molecular markers of treatment resistance have a huge impact when you want to understand how well your therapy is actually clearing the disease from the patient. And these are just a few benefits, and I would argue there are many others to understand this multi-scale heterogeneity. So now that I've hopefully motivated in you that this understanding that multi-scale heterogeneity is important, I now want to talk about a particular case study, which I think is a real poster child for multi-scale heterogeneity in complex disease. And this is a condition that is called facioscapulohumeral muscular dystrophy, or FSHD for short. So what is FSHD? Well, it's a genetic muscle disorder which is inherited in a dominant fashion and is linked to chromosome 4, either genetically or epigenetically. It has a prevalence of about 1 in 7,500, which is quite common for a muscular dystrophy, but still makes it a relatively rare disease. But this prevalence is increasing as we're getting better and better at diagnosing this complex condition. Molecularly, the condition is linked to misexpression of a protein that is called DUX4. And this is a transcription factor, which means that it can activate other genes and cause them to change in their levels. And this is the main therapeutic target for FSHD. But unfortunately, there are currently no approved therapies for the condition, though there are several in clinical trials, some of which do target DUX4. But in addition to the lack of treatment for this condition, there is also no health advice for patients that is really robust and well validated on clinical scenarios. So when a patient is diagnosed with FSHD, it's very difficult for the clinician looking after them to say, this is a way in which you can live your life. This is the sort of exercise you should do. This is the sort of diet you should have in order to prevent progression of the condition. That's not very well known. Clinically, the condition historically is defined as a descending skeletal muscle atrophy, which means that it typically begins in the facial muscles and then progresses to the upper body, and then latterly in later life can have involvement of the lower body, particularly the tibialis anterior, which can result in foot drop and a lot of difficulty in patients walking around. Now, this has been the textbook definition for a very long time now, decades and decades. However, recently, a lot of clinical studies have shown that it's not really the only way in which FSHD can present. And in fact, we showed by this analysis about 700 UK individuals that there might be up to 7 distinct clinical subtypes of FSHD. And we also found that about 1 in 4 patients do not present with this descending skeletal myopathy. Some of them present with a facial sparing myopathy, and some of them actually present with a myopathy that begins in the lower limb. And you can see how important this would be if you're trying to understand clinical outcome measures that are based on how far a patient can walk, for instance, in 2 minutes, which is a common outcome measure for clinical trials in neuromuscular disorders. If you have a set of patients who are predisposed to progress more quickly in the lower limb compared to others, you want to make sure you identify them early so that you can allocate them equally to each arm of your clinical trial. In addition to this diversity in clinical presentation, you also have a diversity in age of onset of the condition, with some patients affected in childhood and often becoming wheelchair-bound in childhood, and other patients living into late life, into their 80s without very much clinical impact of the condition. And we don't really know why this is. We don't know why these 2 patients should have this difference. And indeed, we can look at identical twins, monozygotic twins with FSHD, one of whom will have a very severe disease phenotype with infantile onset, childhood onset of the condition, and another which will have almost no impact throughout their life. And really, this hints at the possibility of non-genetic modifiers for the condition, but we don't understand what these are. And it doesn't stop there. As I mentioned before, in addition to this population-level heterogeneity, you also have heterogeneity in how the condition manifests within a single individual. So to demonstrate this, we can perform whole body MRI on patients and look at their muscles and look at different sequences of the MRI. And we can find sort of 2 key hallmarks of the condition. One is inflammation, as detected here by stir positivity, but it can also be detected by term positivity on MRI. And also we have fatty replacement of muscles, which is detected by T1 positivity on MRI. And you can see here healthy muscles by comparison don't tend to have these sequences. There is a huge diversity within a single individual of which muscles will have inflammation or fatty replacement. There's often, although it's not apparent so much from these images, significant left-right asymmetry. And while we know that fatty replacement seems to be associated with clinical weakness, as you can imagine, having fat instead of muscle will lead to that, the inflammation doesn't seem to have a clear clinical consequence other than it accelerates this fatty replacement that happens in the future. And we don't know what triggers this inflammation and what really leads it to accelerating this fatty replacement. So understanding this diversity in the muscles that are affected in FSHD within a single patient is very, very difficult and hugely important. But on top of that, there's another layer of heterogeneity, which I alluded to earlier, which is this tissue scale level of heterogeneity. Here you can imagine this is a biopsy taken from an inflamed muscle of an FSHD patient. You can see the sort of perivascular inflammation here and endomysial inflammation here around the muscle fibers. Now, if we were to isolate from this biopsy myoblasts, which are muscle fiber precursor cells, and look at the expression of DUX4, which is this major molecular therapeutic target in FSHD, we will see that DUX4 is only expressed in about 1 in 1,000 single muscle cells at any one point in time. This is a hugely heterogeneous distribution of what is supposed to be the major therapeutic target in FSHD. And we don't understand the reasons for this heterogeneity in the distribution. It could be that this small subset, 1 in 1,000 single muscle cells, have something special about them that mean that only they express DUX4. Or it could be, because we've just looked at one time point, that these cells are flashing on and off with DUX4 all the time, and we're only capturing a small fraction of that. But these 2 possibilities have a great impact for the efficacy of a potential anti-DUX4 therapy. And so understanding this heterogeneity at the tissue level is extremely important. So hopefully now I've motivated for you a bit of interest in multi-scale heterogeneity. And I've demonstrated to you that it's a real big problem when we want to understand a complex pathology like FSHD. Next, I'd like to talk to you a little bit about some of the work that we've been doing over the last few years in a very large collaboration to understand this heterogeneity at multiple scales and show that at each scale we can unveil something that can be of clinical consequence and potential use for streamlining clinical trials. So the first thing I'm going to talk to you about is characterizing FSHD at the population level, understanding these interpersonal differences and how they can contribute to disease progression. And the work that I'll be describing here is outlined in our paper that's currently under review, but if you would like to read it, it's available on the medRxiv at the moment. And this is a wonderful collaboration that was performed with Dr. Jeffrey Statland in Kansas University Medical Center, and also Professor Peter Zamet at King's College London, and also Ketan Patel at Reading University. And what we did in collaboration with Jeff Statland was obtain access to this wonderful data set, which is the USA FSHD Patient Registry. And it's a very complex, multimodal data set. that describes about 500 FSHD patients. And when each of these patients enrolled in the registry, they completed a detailed questionnaire that outlined potentially known disease modifiers of FSHD, such as the genotype, here the D4Z4 short array length, and also sex, and then also potential disease modifiers that hadn't been very well characterized in FSHD, such as cigarette smoking, high blood pressure, diabetes, high cholesterol, and body mass to height index, or weight to height index, rather, body mass index. And then what happened is that each of these patients were followed up for an average of 8 years. And each year they then completed a detailed questionnaire describing the severity of their symptoms of FSHD. We then spent quite a lot of time carefully, manually curating these questionnaires to understand the most clinically relevant variables from these qualitative and quantitative outputs. And what we did was we assigned questions to one of 3 muscle group involvements: upper body, facial, and lower body involvement. And we said that a patient had involvement of each of these muscle groups at the point in time when they had submitted a questionnaire answering yes to the most common symptoms associated with that muscle group. And what I really want to highlight here is the variables that comprise lower body involvement. You can see that a patient that is recorded as having lower body involvement has said that they cannot run, they cannot climb steps unaided, they have asymmetric leg weakness, and they have back and hip pain. These are really quite important functional characteristics for a patient trying to live their day-to-day life. So lower body involvement is really quite a severe impairment in this particular data set analysis. The next thing that we did was we performed a time to event analysis for each of these different muscle group involvements separately via a multivariate Cox proportional hazards model to determine the impact of each of our 7 possible and known FSHD disease modifiers and determine their impact on the risk of developing involvement of each of these muscle groups. And this was the first prospective time to event analysis that really investigated how common risk can impact on FSHD symptom onset. Here is the results of this analysis displayed in these forest plots. And what you can see immediately is that the well-known association between short D4Z4 repeat length, which is the genotype of FSHD, and earlier facial upper body and lower body involvement is clearly recreated by our analysis, which gives us some confidence that the methodology is valid. But something that we then saw that has not been demonstrated before was that patients who reported smoking cigarettes at baseline had twice the risk of earlier facial involvement and this important functional lower body involvement compared to patients that didn't smoke. And you can see this quite clearly in these Kaplan-Meier plots for facial involvement and lower body involvement, where non-smokers are outlined in yellow and smokers are outlined in blue. And we can see here for the lower body involvement, at around the age of 50, a patient that reported smoking at baseline has about a 50% chance of this quite severe lower limb involvement, whereas a patient that reported not smoking has only about a 20% chance of this lower body involvement. So quite a dramatic change in the symptom onset. So the next thing that we did was take this clinical insight derived from these registry studies and went back to the lab. And this was work that was done in collaboration with Professor Peter Zammit and also Philip Heher and Ketan Patel over at King's College and Reading University. And what we did with this assay was we obtained myoblast cell lines from FSHD and healthy patients. And we employed this very interesting assay in which you can get a laboratory grade cigarette, pull off an inhalation of cigarette smoke and bubble that through phosphate buffered saline in order to dissolve all the soluble components of cigarette smoke extract into a media. We can then culture these myoblast cells and differentiate them into these muscle fiber precursors, which are shown here with this immunostaining for myosin heavy chain, in the presence or the absence of this cigarette smoke media. And what you can see here quite clearly is that the control individual, this is a female biceps muscle biopsy myoblast that I isolated from, differentiates to form these nice chunky myotubes. And those cigarette smoke media does reduce their size a little bit. It's not that significant. However, in the case of FSHD, where you can see that the myoblasts already struggle to form large, strong myotubes, when you add cigarette smoke media, you really blunt that ability of these myoblasts to regenerate into multinucleated myotubes. And so this takes this clinical insight that we've developed from studying intra-patient heterogeneity and really translates it into a biological inference, which is that when patients are exposed to cigarette smoke, they can't regenerate the muscle as effectively. And we've done a number of other experiments that have linked this issue to deficient mitochondrial function in FSHD. And for full details, you can see this article or talk to me afterwards for sure. Okay, so that's one level of heterogeneity. That's understanding diversity in the clinical presentation of FSHD patients. But there's other levels of heterogeneity that I've discussed earlier. And the next one I want to talk to you about is intra-personal heterogeneity, this diversity in how the disease is manifesting within a single individual. And for this study, we collaborated with Radboud University Medical Center, in particularly Professor Bazel van Engelen, Dr. Anna Greco, and Dr. Leah Houston were really instrumental in the analysis of this particular cohort. And they collected data from a group of 25 FSHD individuals and 35 control individuals. And what they did that really allowed us to understand this intrapersonal heterogeneity was perform multimodal assessments of these patients. So they did lots and lots of clinical assessments, including well-validated clinical scores, such as the Ritchie and Lamperti score. And then they did FSHD muscle MRI. So they did MRI on all the FSHD patients to identify which muscles were inflamed and which muscles were not inflamed in each patient. They then performed MRI-guided muscle biopsies, extracting from each patient a biopsy from a non-inflamed muscle and an inflamed muscle. And for a subset of patients, we also obtained peripheral blood mononucleated cells in order to understand this interaction between the circulating immune cell and the inflammation that is happening in muscle biopsies. We then performed RNA sequencing on all of these samples to get a really detailed multimodal picture of what's happening on an individual patient basis in terms of the diversity of FSHD molecular phenotype. Obviously, we did a lot of different things with this data, so I'll just show you a few things that we found that are quite interesting from the perspective of heterogeneity. The first thing that we did was we performed a simple differential expression analysis, looking to identify the genes whose expression was different between control individuals and FSHD individuals, but also paired within FSHD individuals between the inflamed, what we have here as term positive samples in orange, and the non-inflamed term negative samples in the lighter yellow color. We then performed a k-medioids clustering of these samples based on a feature space that was derived from this differential expression analysis. And we performed consensus clustering and used cluster stability plots to determine the optimal number of clusters in the dataset. What we found was quite striking in that if you look at the cluster distribution here, you can see this red cluster contains only individuals who are control. And actually, there was a considerable amount of homogeneity amongst the muscle biopsies from control individuals. However, all these other muscle biopsies are from FSHD individuals, and you can see that there's considerable heterogeneity in the gene expression landscape in these samples. And some samples fall into multiple different clusters, with many of the inflamed samples forming into little singleton clusters with almost unique gene expression profiles. What we then did was looked at this largest cluster, which contains mostly non-inflamed FSHD samples. We noted that a small proportion of the non-inflamed FSHD samples lay outside this cluster, and these tended to look a bit more like the inflamed samples, which, as I mentioned earlier, were associated with an accelerated disease progression. We then had a hypothesis that perhaps the patients from whom these non-inflamed FSHD muscle biopsy samples were taken, who lay in this green cluster, might have a milder phenotype than the patients whose non-inflamed samples clustered closely with these more aggressive inflamed samples. And this is something that we found to be the case. We found that the patients whose non-inflamed muscles lay in the green cluster had a lower limb fat fraction as assessed by MRI, had a lower clinical severity score, and tended to have a shorter disease duration than those whose non-inflamed muscle biopsies clustered with the inflamed ones outside of this particular study. So this shows you that you can essentially use the molecular profile of muscle biopsies taken from individual patients from multiple sites in those individual patients to understand a little bit more about how we can develop molecular biomarkers for clustering patients into milder and more severe clinical groups. The next question that we wanted to ask is, what are these genes? What are these genes that are able to discriminate control from mild FSHD from severe FSHD? And so we performed a gene set enrichment analysis to answer this question. And what we identified was strong enrichment of genes that are involved in mitochondrial processes and mitochondrial biogenesis. And this was quite interesting in concordance with a study that we performed a number of years ago in human molecular genetics, where we identified suppression of mediators of mitochondrial biogenesis as important for FSHD muscle regeneration defects. Here, we found in this particular data set that the estrogen-related receptor alpha seemed to be suppressed in both non-inflamed and inflamed FSHD muscle compared to control, as did PGC1 alpha And this seemed to result in a suppression of mitochondrial genome expression as ascertained by the expression of genes that are encoded by the mitochondria. And this suggests that suppression of these two proteins that are critical for mitochondrial biogenesis could result in fewer mitochondria in FSHD muscle. This then took us back to the lab because it gives an interesting possible insight. Because estrogen-related receptor alpha, though it cannot bind human estrogen, has a binding pocket that can be fit by plant-derived estrogens, such as those that are present in soy isoflavonoids, including biocanin A. And we found that by adding these biocanin A soy isoflavonoids to FSHD cells, we can actually increase the expression of estrogen-related receptor alpha, which is autoactivatory. But perhaps even more striking was that when you added this biocanin A to FSHD muscle cell cultures and matched controls, we found that while biocanin A had very minimal impact on the ability of the controls to regenerate into large multinucleated myotubes, this was not the case for FSHD, where we saw a dramatic improvement of the regeneration phenotype, as quantified here by increase in myosin heavy chain positive area on these immunostaining. Okay, so this has given a bit of an insight. So by looking at the clinical heterogeneity of FSHD, we can suggest maybe patients should stop smoking. By looking at this intra-patient heterogeneity in FSHD, maybe we could suggest they could eat soy. But if they do either of these things, how do we know if it's helping them? How do we monitor progression in FSHD? And in order to answer this question, we need to develop biomarkers for the condition. And we spent a number of years, by me, I mean me and Professor Peter Zammit at King's, developing a transcriptomic biomarker of FSHD status, severity, and indeed progression based upon muscle biopsy gene expression of a number of the targets of a transcription factor called PAX7. But there is a problem with these particular biomarkers, and that is that they are invasive. They require us to take muscle biopsies from FSHD patients, often serial muscle biopsies. And this is just not something that is acceptable to a patient. These are painful biopsies, and to do them in a serial manner is potentially dangerous in terms of the regenerative pathology of the condition. So we need to develop minimally invasive biomarkers. And we have been quite interested in the idea of circulating biomarkers, i.e. blood-based biomarkers. And this lovely collaboration with Radboud University gave us an opportunity to investigate precisely just this, because we had paired peripheral blood mononuclear samples alongside muscle biopsy samples. And this allowed us to compute the level of the PAX7 target gene biomarker in muscle and look to see how well it correlated with the level in blood samples from the same patients. And what we found was there was a very strong positive correlation between the level of our biomarker in muscle and the level in matched blood from those patients. And in fact, when we refined this biomarker, we were able to demonstrate that measured in blood, it correlated with clinical severity in these patients. Now, we're not entirely sure of the biological reasoning behind why this gene expression in muscle should correlate with blood, but it does seem to be quite a robust empirical finding. And in fact, we validated it on an independent cohort of 54 FSHD patients who had blood samples taken, gene expression profiled, and also severity quantified at the same time. And what we found was, again, this refined PAX7 biomarker correlated very well when measured in blood with the level of severity in the FSHD patients. But what was quite interesting was that we found when we looked at younger patients, this biomarker was fairly modest in its ability to tell apart mild FSHD samples from severe. However, when we looked at older patients, we actually got a much more robust discrimination, which suggests to us that as patients are getting older, the biomarker is becoming more accurate, which suggests that patients are aging into these quantifiers of severity, which gives us the implication that it might be a predictive measure. And this is something that we're investigating now in longitudinal studies. Okay, so now hopefully I've motivated why studying clinical level heterogeneity and intra-patient heterogeneity can give important insights into how we can develop biomarkers for conditions, understand modifiable risk factors, and get insight into the molecular mechanisms of FSHD in order to help us develop therapies. The last thing I want to talk about is this tissue level heterogeneity, this cellular heterogeneity, and its relevance for FSHD. So as I mentioned to you earlier in the talk, FSHD has a major therapeutic target, and that is DUX4. But when we look at cells isolated, muscle cells isolated from FSHD patients, we find that DUX4 expression is only present in about 1 in 1,000 myonuclei within a single muscle cell culture. And so we wanted to develop a model to understand DUX4 expression to really quantify whether this can be explained by DUX4 flashing on and off, DUX4 rapidly killing cells, or whether DUX4 could just be restricted to a small number of myonuclei. So we developed a compartment model that took inspiration from the very old but recently very popular field of epidemiological models, which became very popular during COVID. And essentially, it's quite a simple model where we have a cell occupying one of 5 different states. The first is what we call a susceptible state, where the cell does not express any DUX4 and it doesn't express any targets of DUX4. Now, this is because DUX4 is a transcription factor that activates targets, and therefore the presence of DUX4 targets can, in a sense, be thought of as a proxy for historical DUX4 expression. We then say that this susceptible cell can become an exposed cell once it expresses DUX4 but hasn't yet expressed the targets. DUX4 can then be translated and the targets transcribed to get what we call an infected cell, which has both DUX4 and targets present in it. And then this can transition to what we call a resigned cell if the DUX4 mRNA degrades and we just have target genes present but no DUX4 mRNA present. Now, we previously shown a number of studies that it's really the target genes of DUX4 rather than DUX4 itself that are responsible for driving cell death in FSHD. And so we have a 5th compartment that the cell can be, which is dead. We have a lot of interesting transition rates between these different cells comprised by the expression regime of DUX4. So we have the transcription rate of DUX4, we have the degradation rate of the mRNA, translation rate of DUX4, transcription rate of targets, and the death rate of target positive cells. But we also have this very interesting additional parameter that is specific for muscle fibers. And this is the diffusion rate of DUX4. And this is based on the fact that the muscle fiber, rather than being a single cell like most cells in the body, is in fact a syncytium. It's one large cell comprised of multiple fused cells. So it's a multinucleated cell. And as a consequence, you can imagine that a protein that is derived from mRNA expressed in one of the nuclei might actually be able to diffuse and enter into other nuclei in order to spread a cascade of target gene activation. So we incorporated this parameter. Then, through a lot of careful experimentation performed by Johanna Prula in Peter Zamet's lab and also Matthew Cowley, who's a PhD student at Bath, we were able to experimentally estimate all these different parameters by consideration of inducible DUX4 myoblast cell lines, as well as single cell and single nuclear RNA-seq data. The next thing that we were able to do was express this model as a system of ordinary differential equations with these experimentally derived parameters, and then simulate this model forwards in time. What we found when we did this, so we simulated the model forwards in time from the starting state of all cells not expressing DUX4 in the S susceptible state, and we simulated it forwards in time for 3 days, and then we compared it to real-world data of single-cell RNA-seq and single nuclear RNA-seq from FSHD patients that had been in culture for 3 days. And we found a real remarkable similarity between our model predictions of each of these different cell proportions and the true actual cell proportions that could be derived from these different data sets. But the added value of our model is that instead of what single cell and single nuclear RNA-seq is quantifying, which is firstly a single time snapshot, and secondly, only live cells, our model can be simulated through any number of time points and can also predict the proportion of dead cells that you will get as a consequence of this. Now, this is extremely important for anti-DUX4 therapeutic design, because you can imagine that if we are to develop an anti-DUX4 therapy, we can imagine that it will be modifying one of these parameters that we have experimentally estimated, including the DUX4 mRNA degradation rate, which is one that is currently being looked at by a number of different approaches, including siRNAs and antisense oligonucleotides, as a potential parameter to target in order to therapeutically impact DUX4. But here we've developed an in silico toolkit to allow one to simulate the impact of any of these therapies in terms of their change on the DUX4 expression parameters, and see what this means for cell death and the distribution of cell death within a muscle fiber. And we've developed a suite of in silico tools to really examine this particular gene expression trajectory, but this could be applied to a wide variety of other models. And the paper has actually just recently been published in eLife, if anyone is interested to look at that. So hopefully over these sort of last 30 minutes, I have convinced you that multi-scale heterogeneity is a very important thing to understand, and that it can drive great insights into complex pathology, both from understanding risk factors, both from understanding the molecular mechanisms of disease and designing therapy, to developing in silico tools to understand how well your therapy is going to work, and also from developing biomarkers for refining clinical trials. And the techniques that I've sort of described can be applied to many other pathologies, but there are certain caveats here. And the key caveats are that relevant data is really key. you need data that describes multiple scales. So for the population scale, you need large cohort studies. For the individual scale, you need lots of multimodal assessments of your patients. And for the tissue level, we need more emerging technologies like single cell and single nucleus technologies and spatial transcriptomics. But on top of that, and probably almost as important as the data, is interdisciplinary crosstalk. Because to understand multi-scale heterogeneity, you really need expertise from multiple scales. You need experts in molecular biology, you need experts in clinical medicine, you need experts in cellular biology, you need experts in radiology. You really need a big network. So it requires a lot of collaboration between different disciplines. And I would also emphasize that methodological challenges do really remain when you're trying to characterize multi-scale heterogeneity in complex pathology. And one of the key problems is integration of data across these different scales. How do we go from the tissue level heterogeneity all the way up to the clinical heterogeneity? Is there a way of integrating that data? And that's still quite an open problem, and one that I'd be delighted to discuss if anyone's interested. And with that, I will close and thank all my many collaborators, particularly here at Turing, and also at King's College London, at Radboud University, in Kansas, Rochester, and Bath, and also the John Walton Muscular Dystrophy Centre in Newcastle. And thank my funders, and thank you all for listening. And I welcome any questions, but if you think of a question later, you can also feel free to email me at this email here, cbanerji@turing.ac.uk. Thank you very much for that.
Thank you so much, Chris. That was great. If you just stop sharing your screen, sorry, I've got a bit of a cough. Anyone can ask questions in the Q&A box at the bottom bar. Or if you would like to ask your question live to Chris, then under the reaction bar, you can also raise your hand. Ashish, I can see you've asked a couple of questions in the bar, so I'm not sure if you want to ask any of those live.
Yeah, thanks, Vicky. Great talk, Chris. I don't know if you can, I'll probably just read them out. So I am very interested in cancer. So I was wondering, especially for cancer mets, when it spreads to multiple organs, have you or others applied this kind of paradigm of multi-scale heterogeneity to study cancer mets? And a related question is, this is a rare disease. I was wondering, have you applied it or others applied it to study more common diseases like diabetes and cardiovascular disease? And I had a technical question on one of the slides that you had this number called, 1 in 1,000 nuclei or micronuclei express DUX4. So I was two questions on that. Does it change with the site of the muscle tissue? And does it change with age or disease progression? So yeah, so I peppered you with a bunch of questions, but you can refer.
No, but I welcome them. Thank you. What I'll do is I'll start at the end and I'll work my way backwards. And if I miss one, just let me know. So in terms of the DUX4 expression, the 1 in 1,000 nuclei. So this is generally based on cultured cells. So if you take a muscle biopsy from an FSHD patient and you isolate myoblasts from that and then you immunolabel them for DUX4, then you'll see roughly about 1 in 1,000. The number varies. If you differentiate the cells, it tends to go up slightly, but not to really much more than a few percent of cells that are expressing DUX4. In terms of looking at DUX4 expression,
so my question was, does that number show great variance depending on where you take the biopsy from?
Not really, no, not really. But the problem is really, is that there's not been that many studies that have taken cultured cells out from multiple biopsies from multiple sites. So there's not really been a study that's done that. Most studies from myoblasts tend to be derived from biceps or deltoids, sometimes from traps, and they don't really show that much variation. But the problem is really that it's very hard to measure DUX4 itself in a muscle biopsy. It has to be in these sort of cell ex vivo sort of cells that are extracted from the muscle biopsy and expanded, because it seems to be more that's basically the subset of cells that are expressing DUX4. So actually looking at the biopsy in itself, you very rarely see DUX4. So the answer to that question is no, but it could be that with more data that changes. And in terms of whether it changes with age and things like that, the answer to that is no. And actually I've done a lot of work on DUX4 itself as a biomarker, and DUX4 target genes as a biomarker of FSHD, because they seem to be a bit easier to detect in the biopsies. And there is evidence that the DUX4 target genes, at least, seem to be associated with this inflammatory situation that happens in the STIR-positive and TERN-positive MRIs. But it doesn't seem that DUX4 target genes are particularly well characterized, well characteristic of non-inflamed FSHD loss of biopsy. They're not that good in terms of a diagnostic biomarker or a prognostic biomarker, sorry, or a severity correlated biomarker. And you'll have to remind me of the next questions because I have forgotten.
So applying this paradigm of heterogeneity, multi-scale heterogeneity to cancer mets in particular, and more common diseases like diabetes and cardiovascular disease.
Absolutely. I mean, I'll be honest, I'm not an expert in those areas, but I would imagine from cancer mets, there's a huge amount of scope to apply these kind of approaches, particularly if you're considering more sort of single cell technologies at the moment. If you're taking multiple sort of, if you have mets in multiple sites, for instance, you could imagine characterizing the gene expression landscape of single cells from those multiple sites. There's going to be two layers of heterogeneity. In amongst that, there's going to be the heterogeneity from the sort of evolution of the cancer away from the bulk tumor, I imagine. And then there's also going to be sort of a spatial heterogeneity in the way that it's metastasized to a particular site is probably going to require a particular molecular marker. So I think from that would be quite interesting. And then, of course, the next stage would be to look at the distribution of them across the individual in the same way that we're looking at the distribution of muscle that's affected in FSHD. And I imagine you could do that with sort of CT, MRI sort of scans and link it up there. And then finally, of course, the impact it would have on clinical progression and treatment management. So I think, there's a very clear way of translating this exact sort of approach, sort of lifting it and putting it into the cancer metastasis setting. But what I would say is that you just need well-characterized data. And if you have that, then, and that's something that you work on, I'd be delighted to talk more about those approaches.
Thank you. Enjoyed the talk thoroughly.
Thank you very much. Thanks for your questions.
Thank you, Chris. We haven't got any more in the chat for the moment, so do feel free to add yours. Or does anyone else have a question? You can raise your virtual Zoom hand with the reactions button. If not, I'll give it a minute or so and then I'll ask a host question if not. Okay, I will use host privileges while everyone's having a think. So Chris, I guess, I didn't know if you wanted to share a bit more about kind of how you see the kind of heterogeneity in rare diseases, as you kind of mentioned about many people think they would kind of be less heterogeneous, but like you mentioned, kind of on that data. So I didn't know if you wanted to expand on that point.
Yeah, sure. I mean, I think it's quite a concern, heterogeneity in rare diseases, and it's one that we've really noticed in the FSHD field. And really, I think initially it's related to a paucity of data. So heterogeneity is something that will emerge when you have sufficient data to see it, right? So if you can see sort of diversity in clinical trajectories, you see diversity in molecular profiles. And I think for a long time in FSHD, it was something that we could only really see because we didn't have very many samples. When you looked at, for instance, transcriptomic data for FSHD patients, you would see, if you tried to do a clustering analysis of them, you would see something similar to what I presented here. A huge number of little clusters that cluster far away from a tight cluster of control samples. And this is kind of an indication when you have a small number of samples that you might have quite a heterogeneous condition to work with. And it was our sort of first sign. And since then, a huge amount of data has really been collected on FSHD, sort of both clinically, molecularly, and cellularly. And this has really shown us that there's a huge amount of heterogeneity there. And this really presents you with a huge problem, I think, in a rare disease. Because if you have a lot of heterogeneity in your rare disease, you start thinking about stratification for clinical trials, lots of different variables to control for, but you don't have that many patients to run the clinical trial for. So this then becomes an issue of a low number of samples and a huge number of variables to control for. And it's a big issue in rare disease, and I'm sure it's one that's faced in other rare diseases. And I think it sort of motivates a better understanding of how we can control for these sort of heterogeneity features within the context of a small sample in a clinical trial.
Definitely, thank you for expanding on that. I will just give it a couple more minutes to see if we have any last minute questions. If not, as I think we mentioned at the start, Chris is also in our Slack workspace, which again, I'll post a link to. So if there is anything you want to ask him, or I know he's very keen to connect with people. So we also have that option as well.
I did have a more general question. I'm sorry, I was a bit late to the
No,
meeting.
that'd be great.
Yeah, I just wondered what you think the benefits might be bringing in kind of more genetic testing in the NHS. And I know cancer treatment has been talked about already, but I know there can be genetic tests that help identify cancer treatment that might be more effective from the get-go. So rather than using a kind of trial and error approach, testing one and it didn't work and then trying another, could go straight to the genetic testing, and it might help people's health kind of more efficiently. But I think maybe the reason they're not doing that so much is for cost reasons. I don't know how we kind of argue that cost kind of risk benefit. Obviously I see the benefit in it, but yeah, just
I also see the benefit in it, of course.
Yeah.
I think it's a very it's a very sort of important approach, because there's obviously going to be modifiers of therapy, and there's going to be lots of genetic modifiers, and particularly in the context of malignancy, but also in other more common pathologies, things like diabetes and things like that have already been mentioned. And I think that the cost of sequencing is coming down.
Yeah,
but probably still a bit prohibitive to roll it out in an NHS context. And so I think in order to do that, it needs maybe some more sort of clinical justification of how it will improve and reduce morbidity and mortality. And I think from the sort of approaches that I've been describing today, sort of these in silico approaches where you can sort of simulate, if you have this modifier, it will improve the efficacy of this therapy, or this synergistic approaches and things like this. That can really be approached with these sort of in silico models. But also, sort of general, if you have a detailed characterization, I know there's large genome-wide studies, things like 100,000 Genome Project and things like that, where you have a lot of sort of clinical data associated with genetic data. And if that can be mined using sort of ML approaches and things like that to really understand the impact of particular genetic modifiers on survival in cancer, I think that could provide a real strong motivation for and cost benefit analysis really for sort of introducing more routine genome-wide sequencing in particular contexts.
great, thank you. And do you see this kind of like personalized medicine approach being rolled out across not only physical health, but also mental health and more kind of neurological conditions such as dementia and Parkinson's treatment?
I do think so, but I think again that there is a challenge there, and that challenge is sort of paucity of data and reliability outcome measures, and I think that can be quite challenging in a lot of these different circumstances. And I think there's often issues with Alzheimer's as well, in terms of the definitive diagnosis is often one of post-mortem. You can enroll people who you think have Alzheimer's clinically, but might not actually end up having pathological Alzheimer's, and that can lead to sort of difficulties in these personalized medicine approaches. So I think it's a more challenging area, but I think the same sort of tools can be applied if the correct data is collected, I think.
Thank you.
Thank you so much. We've got another question in the chat that I will just read out now. So as you mentioned, there is something non-genetic impacting the manifestation of FSHD, and you mentioned the impact of cigarette smoking. Can you say more about what other factors might be and how they may be identified?
Can I say what the other factors might be? Not definitively, really, because I think we need to do larger scale studies to really understand what the modifiers could be. There's a lot of interesting data that is coming out from different sort of cohorts. So most of the data on FSHD has been collected in a Western cohort, so either Europe or America. And over the years, there's been more data that's come out of sort of East Asia, so Japan, and more recently data is coming out from China, and I know they collected data in India that has shown that in these, particularly in Japan, and I think also in China, it's starting to become more clear, the genetic aberration that causes FSHD has to be more severe in order for you to get a clinical phenotype in these Eastern Asian populations. So this suggests that there might be some modifier in these East Asian populations compared to the Western populations. And whether this is environmental or genetic is not clear, but could potentially be answered a bit more clearly with migration studies. So I think this is something that really needs to happen in FSHD. It's sort of the classical studies that were done in breast cancer, you know, where they found that there was a lower prevalence of breast cancer in Japan, and then it went up a little bit in Hawaii, and then it went up again in migrant populations who went to California, and this was sort of showing that there was an environmental risk factor for breast cancer. And I think now that we're seeing this discrepancy in terms of the clinical phenotype in FSHD in East Asian populations seems to be milder for the same genotype, and is more severe in Western populations, we need to look at the migrants. So we need to look at people that have who have FSHD, who've maybe gone from Japan to Western Europe or to America, and look to see whether their severity changes. And that will give us an idea about whether there are environmental or genetic modifiers, and that will allow us to sort of explore those in more detail.
Great, thanks, Chris. I'll again just pause. Oh, yeah, another one's just popped in. Do we think there's hesitancy from participants and patients in relation to privacy when it comes to genome testing? Do you think public opinion to this will change? I realize that's quite a big question for you, Chris, but I don't know if you have any thoughts on
that. You know what, I think that there definitely is hesitancy among some groups for sort of more widespread genetic testing, and I think, you know, not without reason. But I also think, you know, there's a lot of benefits that can be had from it, and I think it's very much a personal choice as to how it's changing. I don't know, because I've not done those studies myself, but you know, it's not something that I can really clearly comment on. But I would imagine, I would hope that it would change as sort of the benefits of this sort of thing comes out. But the problem is that alongside the benefits of understanding how genotype can impact severity of your conditions and things like that, if you suffer from long-term conditions or even short-term ones, alongside that, there's often you know, a lot of examples of, you know, data privacy being violated, and, you know, a lot of hacking and things like that, particularly in large industries. And I think as long as they exist, as long as those stories exist, as long as that becomes a possibility, there will always be concerns over data privacy. And so I think we need to evolve, alongside of collecting more data, we need to evolve better methods for ensuring that we have absolute security of the data alongside that.
Great, thank you. Yeah, just a bit of a funny one for us to maybe to finish on, but really useful discussion. I think I'll draw us to a close there just to keep us on time. But thank you so much for joining us.